we have made it to our last section, our last major group of organisms that we see evolving on our planet. So let's think about how far we've come. We talked about how the first cells formed. We talked about how the first eukaryotes formed. We talked about how plants moved on the land. We talked about fungi moving onto land. Now, this last group, animals, here we go. Okay, how did animals come about? What is their evolutionary history? What's their story? Before we jump into their story though, let's describe first what is an animal. Let's start out with the basic definition of what are the derived characteristics of organisms that we call animals, right? So all of animals are in their own kingdom, okay? So it's a monophyletic group. We characterize them. Hang on, let me take care of Greta. Okay, how do we characterize animals? Well, animals are chemoheterotrophs, right? So that means that they eat other organisms to get both their carbon and their energy source. They're multicellular eukaryotes. Okay, so they have eukaryotic cells and they're multicellular, unlike protists. But the difference between animals and plants, because plants are also multicellular eukaryotes, right, is that animals do not have a cell wall, right? So they don't photosynthesize and they don't have a cell wall. Animals are some of the only organisms that can move using muscle and nervous tissue, right? So muscle and nervous tissue are unique tissues that we only find in the animal group that allow them to move their bodies around. And they reproduce sexually, okay? So they go through the process of making gametes. Those gametes fuse to make the next generation, so they reproduce sexually, right? So at the core of it, that's really what makes an animal different from these other groups of life that we've been talking about. So when did this all happen? When did animals get on the scene? So plants, we talked about plants moving on to land about 500 million years ago or so. 270 million years before that, when life was still hanging out in the oceans, there really wasn't much going on land. Plants haven't even started to even think about that yet, is when we find the first animals. So some of our early fossils date back to about 700 million years ago. So, right about here, okay? At the end of the Proterozoic era is where we find the first evidence of animals, okay? And they were exploding in the oceans. There are many lineages of animals that were around at this time that just went crazy, rapid diversification in the oceans, that just, those lineages just didn't survive in today, okay? So there's many other groups of animals than the ones that we will be investigating over the next couple weeks that are just not around anymore, okay? We're going to focus mostly on the extant phyla for most of our rest of our term together, but they were going crazy in the oceans at this point in time. So let's take a sense of where are animals in our eukaryotic tree, right? So remember, this is our tree of eukaryotes. All of these in yellow here are protists. Here we have our plants over here in Archaea placida. Animals are over here in this group. They're over here with the fungi in the Uniconta group, right? So Uniconta, cont meaning tail, uni meaning one, so that means that their cells are, especially, well, we see this in the flagella, especially of sperm cells, have only one tail. And within the Uniconta, they are part of the episthecon, which means rear-facing, okay? Or rear-facing tail, rather. So they have a tail on the backside of their cells, essentially is what that means. But what is the closest sister taxa to animals, right? What is the closest sister taxa to the animal kingdom? Choanoflagellates, right here, right? Protists. So we share a common ancestor with these choanoflagellate protists. That gives us a clue about some of the evolutionary history of animals. So what's a choanoflagellate? Here's kind of what they look like. So choanoflagellates are tiny protist cells with many flagella and a ring of cilia coming off the back of their organism, right? So episthecon's back, right? And they can form kind of these little colonies where they have kind of this branch that they sit themselves up on, and then they have these, what we call these collar cells and a flagella. So this arrangement, these collar cells, this is what's unique to choanoflagellates, and we also see this appear in some of the animals as well. So So this collar cells and these flagella, so this collar actually, and this flagella are unique characteristics that they use for feeding. So basically what they do, that collar secretes mucus, and then they use the flagella to basically move water through, and then they trap food in their collar, in the mucus of their collar, and then they eat it, basically. Okay, so these are single-celled or colonial protists, typically, but we see them, and they give us a little bit of a hint as to maybe how did some of the first animals arise. Because we see collar cells in animals too, right? We see collar cells in sponges, which is a phylum of animals that we'll talk about later. We see collar cells lining the cavity of sponges. We see collar cells in some of these cnidarians and platyhelminthes and echinoderms. So this basic structure has been retained by some of the descendants. Okay, so again, parsimony tells us it's most likely that these organisms and all of these animals inherited that characteristic from the common ancestor. So remember when we first talked about how multicellularity evolved in some of the early eukaryotes and that process, right? Kind of the same idea here of these choanoflagellates, these unicellular collar cells, forming a colony and then forming into multicellularity where they can specialize, right? So here we see a single cell becoming a colony and then forming an aggregate with specialized multicellular portions to its body. There's a hypothesis that these may have been kind of floating around on the ocean floor, scavenging food off the ocean floor. And then a mutation arose that allowed them to kind of invert themselves to create a specialized gut area, specialized cells, if you would, not a true gut, it's an organ, but specialized region of cells that are really good at digestion. And we actually have documented this in choanoflagellates are around today. They do this, we see this process of this inversion. And this shape, this ball of cells and then this inversion, we see this in animal embryos, which we'll touch on later in animal development. But this trap, this kind of inverted shape, would have made it easier to trap food, okay? And eventually, we think, this mutation of creating that kind of U shape would have given rise to some of the first digestive systems in animals. So what did early animals look like? We probably wouldn't recognize them today as animals, right? The oldest fossils, again, are around 700 to 710 million years old. And they kind of look like this, kind of like a blob of repeating units. So animals, the early animals, we call them Ediacarans. A lot of them are generally like this. They're soft-bodied, they're small. We don't see any specialized tissues, but those also wouldn't have fossilized very well. But we think that they probably just absorbed their food from the environment, right? Just using basically diffusion to get their food. And they stayed pretty small, okay? We think though that the animals in like Dixonian that arose during this era were probably the ancestors of early sponges and cnidarians, jellyfish, things like that. But for the next 150 million years after this, animals stayed pretty small. And then we see a huge rapid diversification and we basically see these guys disappear. And that's what we're going to go next, is talking about how did most of the major phyla of animals evolve and what happened during the Cambrian explosion.